Welcome to our little curious podcast, Hidden Synapses. What if the secrets of life were hidden in a cornfield? Barbara McClintock saw something no one else did: genes that could move, rewrite themselves, and influence evolution. But her discovery was so radical, it took decades for science to catch up. This is the story of the woman who changed genetics forever. Barbara McClintock's journey from a curious young girl to a groundbreaking geneticist represents one of science's most remarkable stories of persistence and vindication. Born in 1902 in Hartford, Connecticut, McClintock defied the social norms of her era through her fierce independence and intellectual pursuits. Unlike many young girls of her time who were expected to focus on social activities and domestic roles, she preferred solitude and academic endeavors. Her mother's progressive mindset enabled her to pursue higher education at Cornell University, where she ultimately earned her PhD in botany in 1927, a significant achievement for a woman of that time. At Cornell, McClintock's fascination with cytogenetics, the study of chromosomes and heredity, led her to focus on maize as a genetic model. This choice would prove instrumental in her future discoveries. Through meticulous observation and analysis, she began mapping corn chromosomes, establishing herself as a pioneer in the field, and developing techniques that would later revolutionize genetic research. The 1940s and 1950s marked the period of McClintock's most revolutionary work: the discovery of transposons, or jumping genes. This finding fundamentally challenged the accepted notion that genes maintained fixed positions within the genome. While studying maize kernels, she observed unusual color patterns that defied traditional genetic explanations. Through careful microscopic analysis, McClintock identified DNA segments that could relocate themselves within the genome, influencing the expression of other genes. This discovery was particularly remarkable given the limited technological tools available at the time. Despite the groundbreaking nature of her discovery, McClintock faced significant obstacles in gaining acceptance from the scientific community. Three main factors contributed to this resistance. First, her ideas were simply too advanced for the 1950s, when DNA's structure had just been discovered and the field of genetics was still in its early stages. Second, her preference for working independently and reluctance to promote her findings limited their exposure. Third, as a woman in science, she encountered persistent gender bias that often excluded her from important scientific discussions and mainstream institutional support. One of the most telling quotes from McClintock herself reveals her unique research approach. She described her work as listening to the corn, emphasizing the importance of careful observation and intuition in scientific discovery. This patient, methodical approach eventually led to vindication when molecular biology caught up with her theories in the 1970s. Scientists confirmed that transposons exist not only in maize but in all living organisms, including humans, playing crucial roles in evolution, immune system function, and various diseases. The scientific community finally acknowledged McClintock's contributions in 1983 when she was awarded the Nobel Prize in Physiology or Medicine, becoming the first woman to win this award solo in that category. The recognition came more than 3 decades after her initial discoveries, demonstrating how far ahead of her time she truly was. Even after receiving this prestigious honor, she maintained her humble approach to research, continuing her work in her small laboratory at Cold Spring Harbor. McClintock's personal life reflected her dedication to science. She never married, choosing instead to focus entirely on her research. Known for her intense concentration and preference for solitary work, she often delayed publishing until she was completely confident in her findings. This perfectionism, while sometimes limiting the spread of her ideas, ensured the accuracy and reliability of her research. Today, McClintock's legacy extends far beyond her discovery of transposons. She is considered a founding figure in epigenetics, the study of how genes can be regulated without altering DNA sequences. This field has become crucial in understanding cancer development and advancing biotechnology. Her work demonstrates how scientific breakthroughs often require not just brilliant insight, but also the courage to challenge established beliefs and the persistence to defend unconventional ideas in the face of skepticism. The story of Barbara McClintock serves as a powerful reminder that scientific progress sometimes requires decades for revolutionary ideas to gain acceptance, and that the most significant discoveries can come from researchers working outside the mainstream of their field. Her journey from misunderstood maverick to celebrated Nobel laureate illustrates the importance of supporting diverse voices and perspectives in scientific research and the value of persistence in the face of institutional skepticism. Barbara McClintock listened to the language of genes before anyone knew how to hear them. Today, we know that her discovery of jumping genes reshaped our understanding of DNA, evolution, and disease. She waited patiently for recognition, and now, her legacy jumps through every cell of life on Earth. Thank you for joining me on this journey through Barbara McClintock's life. If you enjoyed this episode of our podcast, Hidden Synapses, don't forget to subscribe and share. And if there's another underrated scientist whose story you think should be told, let us know. We'd love to explore it in a future episode.